All right, let's jump right into an incredible explainer on a recent study by Pandya and colleagues that is fundamentally flipping the script on how we approach early cancer detection. For decades, the medical community has been in this massive race to find cancer earlier and earlier. But what if we've been hunting for the completely wrong thing this whole time? This research offers a massive paradigm shift that could literally change how we intercept one of the world's most fatal diseases.
I mean, just imagine looking half a decade into the future of a patient's health. We aren't talking about detecting a tiny early-stage tumor here. We're talking about identifying the precise biological conditions that will eventually allow a tumor to grow in the first place, a full 5 years before a doctor ever makes a diagnosis. It sounds like science fiction, right? It's a bold claim, but as you'll see, it's grounded in some absolutely rigorous science.
So at the absolute heart of this research is a single pivotal number, 14. Out of nearly 3,000 different plasma proteins floating around in the blood, the researchers managed to pin down exactly 14 that carry a unique predictive signal. To find this needle in a massive biological haystack, they used a super advanced machine learning algorithm called XGBoost, along with SHAP-based feature elimination, to really drill down and isolate these specific biomarkers.
Now, what makes this discovery so incredibly robust is the sheer scale of it. They didn't just look at some small, localized trial. They analyzed blood plasma from over 48,000 participants in the UK Biobank, which included 375 incident lung cancer cases. And get this, by combining those 14 proteins with just 4 basic clinical variables, things like age, smoking status, pack years, and COPD history, they achieved a highly predictive accuracy, an AUC of 0.865. That is huge. And they didn't even stop there. They validated this signature across 8 independent external cohorts. They even proved it works perfectly across 2 entirely different testing platforms, Olink and SomaScan. That kind of cross-validation is a massive deal in proving this signal is actually real.
Okay, this brings us to the core paradigm shift of the whole study. The old approach to early detection, well, that's all about finding the seed, hunting down mutant tumor cells or the tiny bits of DNA they shed into the bloodstream. But the Pandya approach is entirely different. They are detecting the soil. These 14 proteins don't actually come from the cancer itself. Instead, they reflect a tumor-promoting environment in the lung. It's literally a signal from the surrounding neighborhood that is primed, inflamed, and ready to support cancer growth.
To really wrap our heads around this soil concept, we need to talk about CAC, or the alveolar transitional state. Think of CAC as a stressed-out panic phase for your lung cells. Diverse, normally healthy cells, whether they're basal, club, or neuroendocrine, sort of converge into this highly specific CAC state when they are under severe stress. And this state creates a very toxic, inflammatory microenvironment. It's this exact environment that acts as the perfect incubator for EGFR-driven lung cancer to take hold.
So the absolutely crucial takeaway here is that this 14-protein signal originates from healthy-looking cells that are just reacting to injury. It is not coming from malignant tumor cells. The researchers traced the biology back and found the signal is sourced from wild-type epithelial, myeloid, and fibroblast cells in the surrounding tissue. It's essentially the lung's healthy neighborhood pulling the fire alarm because the conditions have become dangerously toxic, specifically driven by things like oncogenic EGFR clones.
The chain reaction they mapped out is just fascinating. It's like a biological domino effect. It all starts with an insult to the lungs. Maybe that's particulate matter from air pollution or those EGFR mutations. That insult kicks off a specific type of inflammation driven by a molecule called IL-1 beta. That inflammation then forces the massive expansion of that stressed-out Casey state we just talked about. And finally, those stressed cells release our 14-protein signal right into the bloodstream.
Now, understanding how this biological mechanism works is great, but its true value is taking it from lab theory to actually intercepting cancer in humans. And that brings us to their brilliant retrospective reanalysis of the CANTOS prevention trial. The CANTOS trial originally tested a drug called canakinumab, which conveniently targets and blocks that exact inflammatory IL-1 beta molecule we just saw in the domino effect. So the researchers went back to this trial's data to see, hey, could our 14-protein signature have predicted who would actually benefit from this drug?
And the results gave us this incredibly important number, 55. By using their 14-protein score to flag the highest-risk patients, the ones with a specific inflamed soil, and then treating them with that anti-inflammatory drug, the number needed to treat to prevent one single case of lung cancer dropped to approximately 55. This is a game-changer because it directly solves one of the biggest hurdles in cancer prevention, figuring out exactly who needs the treatment rather than just treating everyone and hoping for the best. Now, it is totally worth noting that a few other concurrent studies, like Zhao's 3-protein model, Liang's trajectory modeling, and Bardwaj's 4-protein tool, have also pinpointed similar proteins using the UK Biobank. Honestly, that's great, because it completely reinforces the biological validity of our 14-protein signature. But what sets the Pandya study completely apart from the pack is their end-to-end translational design. They didn't just stop at a computer prediction. They took the whole shebang from machine learning discovery to causal experiments in mice, all the way to clinical actionability in a human trial.
All right, let's do a quick reality check here, because science is never perfect. The strengths of this paper are undeniable, especially the rigorous machine learning that prevented data leakage and those causal experiments in mice. However, the limitations are pretty notable. For one, the interaction p-value in the CANTOS trial wasn't statistically significant, so the clinical payout is still more hypothesis-generating than definitive proof. Also, this signature pops up in other lung diseases like IPF and COPD, which means it kind of acts as a broad lung injury alarm, lacking total specificity for cancer. Plus, right now, they only have relative protein levels, not the absolute hard clinical cutoffs a doctor would actually need to order a definitive test.
So, what is the actual path forward here? Well, before this becomes a routine blood test at your annual physical, science needs to tackle a few urgent steps. The absolute biggest one, we need a prospective, biomarker stratified trial. Basically, a dedicated study that randomizes high-signature individuals to anti-inflammatory therapy to definitively prove, once and for all, that it stops the cancer from taking root. We also desperately need to build standardized clinical assays with established cutoffs. And critically, this has to be validated in never smokers and diverse populations, especially since EGFR-driven lung cancer disproportionately affects never smokers in East Asian populations, which is very different from the predominantly European, high-smoking cohort they looked at in the UK Biobank.
To wrap up this explainer, I want to leave you with a conceptual reframing directly from the researchers themselves. This signature is, and I quote, a signal of the inflammatory environment that precedes cancer, rather than cancer itself. It forces us to ask a really profound question. What if we stopped endlessly chasing the cancer seed and instead simply fixed the soil before it could ever grow? It is a thrilling glimpse into the future of molecular cancer prevention, and honestly, a massive step towards stopping the disease before it ever truly begins.